So hello, everybody. After lunch, we've eaten. We have much more energy. So hopefully things are going to move faster. I will speak faster, and then I will show even more equations. No, I try to limit this to some extent. Again, interrupt me at any point if you think that I'm either not making sense or I'm flipping these slides too quickly. What I would like to do now, I would like to sort of follow up on some of the things that I told you already in a slightly different context using different software. So what we have learned, hopefully out of this first part, is that there is this software EdgeR, or Edger, some people call it, and we have learned the basic idea behind it related to this GLM and over dispersion. We learned that it uses different ways of measuring or adjusting tags or these transcripts for different dispersions. And then with all of this combined, it has a method of basically estimating parameters, which in our world turns out to be equivalent, in this example that I was showing you, to basically calling things differentially or not differentially expressed. Now we will go a little bit further, because maybe the title of this talk should be Beyond Differential Expression, or something like this. Because really what I would like to talk about now is there are many circumstances, and Soledad Fernandez, the person who was the first speaker this morning, she, I think, gave you some ideas about how statistical design plays into many of these experiments that we are analyzing through various types of omics. So if you have an experimental design and you have a particular types of questions that you would like to ask, then very often it's not just simply the matter of trying to call genes which are differentially expressed, but you specifically would want to identify a subset of your genes or transcripts or other types of omics that are responsible for a particular feature or particular condition in your data. And this can be very subtle. This can be based on the number of different conditions. This can be an attempt to measure some kind of stable factor that differentiates between multiple conditions, or is an effect of time that you are interested in understanding when you're looking at the progression of something. Very, very often we forget about it, but in the omics data models, we are looking at systems, these molecular systems, which are very dynamic. So the whole idea of a gene network, that really doesn't exist something like a gene network. What exists is this constant connection of various switches which are turned off and turned on depending on what's needed, right? So the system is dynamic and it progresses, and we are sort of observing the results of these various changes as the system evolves. Cancer, it's a dynamical process, right? There is basically a growth involving, there is some kind of microenvironment involved in building these cancerous cells, and there is progression of the disease, there is metastases, there is an immune response. All of these things are coming together. So basically, what we're talking about here are very, very small snippets of time at which these systems are observed. On top of the fact that these systems themselves are very, very complicated, we also are only looking at these processes as very particular moments, you know. So I bet you, I shouldn't be really betting, but I could, that if we took your gene expressions measurements before lunch, and then we fed you whatever we did, it wasn't very good. I think it was okay. But, and you do it again, you will have a completely different gene expression, okay? Just because of the fact that we went through this process of a meal. So think about it, because this is part of a challenge. We are basically looking at really noisy systems that are changing on minute-by-minute basis. And we're trying to say something very deep about the molecular principles upon which these things are running. So that in itself is it's just a formidable challenge. And that's why these statistical methods are always attempting to be flexible, in the sense that we're not always sure whether this is the best method, but we kind of can agree that under most circumstances that we can think of, it's not going to do a horrible job, okay? It's not going to be horribly wrong. Sometimes we might be able to find something that will work better. So the things that I will talk about today for the last lecture, are basically what I would call beyond gene expressions, but really it's an attempt to show you a more involved and more complex model beyond what I was talking about this morning. And this model, again, comes out of the group that has sort of looked at the notion of GLMs, but originally, at least, in the context of microarrays. So this model that I will talk about now was originally developed in order to be able to make sense out of microarray data. It was a nice model, and in some sense, it's an obvious model to work with if you already have this background from this morning about tag-wise analysis. So what you should remember, beyond the fact that it's really cool to use HR and it generates nice pictures, is that really the essence of this HR method is that we are looking at things gene by gene by gene, or tag by tag by tag, but somehow we're also taking into account the overall pattern of these genes. And that gives us lots of additional abilities to make right calls. If we didn't use this overall pattern comparison, we would probably not be able to successfully find the majority of the things that we're interested in. So HR was developed later. Lima was actually developed first. But the principle is really very similar. So you can think about this method or this general software piece called Lima as a way of trying to run multiple regressions where you were running regression on each single individual gene. But then somehow you're using the overall patterns of expression of the genes to inform your regression in a similar manner in which we did it in HR. Here, in order to accomplish what we want to accomplish, we'll use Bayesian statistic, or at least some forms of Bayesian statistic. And again, I will only talk for about half an hour now to give you an opportunity to play with some of the software just to see how it runs. But I would like to give you just the basic formulas and the basic formulation of the problem, so you would know that this really isn't a black box, but there is actually lots of statistical considerations that go into this. And there's many people who worked on this for a number of years in order to develop these methods to the point that they are right now. For us, very often, nowadays, it's just pressing a button and it runs, and it basically gives you these odd statistics. But it really is something that lots of people have thought about and tried various different versions of, and then, at the end, they settled for something like this. Again, not necessarily always because it's right, but because it seems to be the least wrong out of the methods that we have available. Okay? So that's kind of the caveat which we always have to struggle with whenever looking at these molecular models. So testing for differential expression. So first of all, I told you last time what the differential expressions are. I actually didn't define it. I told you, well, differential expressions means that the abundances are different, maybe. But at the end of the day, I got out of this by telling you, hey, I have this way of calculating the p-values, either by exact calculation based on this binomial model or based on the approximation using chi-square, and then I would just rank my genes by p-values. The ones that had the lowest p-values would be the ones that I would possibly use as the ones that are differential expressed. I would like to make a little bit more scientific statement about this. Because in general, as you guys can imagine, you probably know this from your earlier elementary statistics courses, there's two types of things that can go wrong when you're trying to discover new things. You can either claim that something is really new when it isn't, or it is something that is important when it isn't, or you can miss something which is important, basically claim that it's not, when it really is. So these are two types of errors that we have to take into the analyses that we were talking about earlier on, and somehow in this context, we have to deal with differential expression. So I will talk about this a little bit and give you some ideas how one can try to use this notion of what is really important, or basically, more precisely, what is the rate of false discoveries. Because at least at the very basic level, what we want to avoid, if you remember my last picture on the slide that I was putting up, and maybe the last one that you saw when you were running our scripts in HR, you have had two examples. One was Poisson example. The other one was the example from the tag-wise analysis with the shrinkage. And on the first one, you had these sea of red. Everything was red. Okay, and on the other one, you had a little bit more modulated response. So only some things were turned out to be red. These were basically, by the way, exactly the same data. So what you should be concerned about is that in the first picture, when everything was red, I potentially marked lots of things in this red color as possibly important, which were not. So that means that I have created a huge false discovery rate. So lots of discoveries in this first picture were probably not true. So I would like to be able to control for this and somehow be able to meaningfully say how many of these red dots, among how many of these red dots do I believe I have false discoveries, and is there a way for me to do some kind of cutoff of these differential expressions in order to keep this in line? So I will talk about it. So the false discovery rate really means the following under what you're looking at the things that are so you have some level at which you will consider something a discovery. So you would basically have tags which are below a certain level. You would say these are the ones that are definitely differentially expressed. So this is your discovery. And false discovery rate really, in some general sense, tells you what proportions of these discoveries are accidental, which means that they really are not true discoveries, but they just came out this way because you're using statistical methods to make the determination. Okay? All right, so this long introduction, let me just... So generally speaking, you can think about gene or tag being differentially expressed by thinking about its probability distribution. So if you basically remember, I was talking about Poisson distributions or some kind of inflated Poisson negative binomials. And I was saying this negative binomial looks like this under one condition and looks differently under different conditions. So that basically means that these two Poisson negative binomial distributions or Poisson distributions, they are really different under these different conditions. So that's one way in which you can define differentially expressed genes. There is a weaker definition, a little bit. So this is sort of, a more detailed definition, but you can also define it simpler, which would include this by saying that, on average, this gene is going to have a higher average in this condition and the lower average in the other, and it also implies that it's differentially expressed. Okay. So if you have a hypothesis that a particular gene or tag is not differentially expressed, then you can use some statistic, some calculation based on observing the threshold, the quantity of that gene under different conditions to try to test that hypothesis. This is basically what we do in statistics all the time. Let's call this statistic Ti, and let's assume that it only depends on the observation of the gene I. So this is okay, but as you can imagine, again, I am going back to my earlier lecture, this will only tell you something about the particular gene, and it's sort of, in a sense, it's throwing away all the other information about anything and everything else that's happening with your other genes or tags. I told you already earlier that we believe that in any given time, there is only a small percentage of genes which are expressed. So by the sheer fact that you're looking at the expression of all the other genes, you have some idea about how the gene that you're interested in compares to all these other expressions, and that already carries information about whether that particular gene might or might not be differentially expressed. So if I just apply this simplistic approach here, which is basically equivalent to this tag-wise approach that we talked about earlier, then I am sort of definitely not taking all the information into account. So there's many different ways in which you can try to adjust for what other genes are doing. This is something which, again, in the microarrays literature and in the early days of microarrays analysis was something that was, and still is, very popular. This is called the volcano plot. And what it does, it basically looks at the p-values. Fortunately, by now I told you how you can calculate p-values under at least one particular method versus looking at the fold changes. So fold change is just a straight analysis of the ratio of expression of tag under different conditions. So this is something which is a volcano plot. And you could say, okay, so if I have things that have very, very small p-value, so this is negative, so basically things which are high up here are really, really tiny in the world of the p-values. So I maybe could be looking somewhere over here if I just look at the p-values. But if I want to look at fold changes, then maybe I would be looking here and here. And if I really want to make sense out of this altogether, then I could potentially apply both of these criteria simultaneously, and I would end up with something like this. So depending how much of the information do you really want to borrow and in what way, you may end up with different numbers of discoveries or different numbers of these genes or tags that you would at the end decide are differentially expressed. So all of these are good ideas. We would want to be able to develop some unified framework that would be better than eyeballing. Because one of the major problems in the plots like this is that we don't really know how to slice this picture. So we don't really know how to put a threshold. You know, I used this line here and this line here, but was this the right line? Or what would happen if I just made it a little bit lower? Well, that means I would have more discoveries. Would these be the false discoveries? So statistics really is needed in order to be able to make sense out of how do you extract these points of your data, which are true signals, and separate them from noise. And you have to do it in such a way that in some sense you wouldn't have too many of these signal points being false positives. Okay? That's the general idea. And again, we need some kind of framework to do this. We'll show you how to do it in software in a couple minutes. In software, it's really easy. You just press a button. But trust me that before you actually can press this button, there was lots of people who were trying various different buttons, And then sometimes it worked, sometimes it didn't. And at the end, they ended up with something that most of the time seems to be OK. So tag-wise, gene by gene, we will now go back to, again, I told you already that this is software and the ideas that were developed for microarrays. So for microarrays, under many circumstances, it was convenient to think about your signals and your gene expressions in terms of continuous variables. Because if you remember the way in which microarrays and then many of these fluorescence-based methods are working, what you're really measuring, you're measuring the amount of brightness of the fluorescence reads on the probes. Okay? So this fluorescence is naturally lends itself to the analyses in terms of some kind of log scale. But generally, it's a continuous variable. It's a non-negative continuous variable. So the type of analyses that I was talking about in terms of counts and in terms of, for instance, RNA-seq data directly, really doesn't apply, at least seemingly doesn't apply immediately. So what we are interested in, we're interested in some kind of level of expression of a gene that is now a continuous variable. I have two different conditions. I can build this statistic, which in sort of standard statistical analyses is something which is called the Ttest. And based on just comparing the differences between the means of the expressions of particular genes under two different conditions, I have some notion of how they differ. And then I can run a formal statistical test in which I would say, well, is this difference on average 0 or not? If it is on average 0, then I would claim that these are not differentially expressed, really, statistically speaking. If it is not 0, then I would claim that it is. So one thing that you guys should realize is that in this kind of analyses, the denominator really matters. Because all of a sudden, I am not just taking a straight difference, but I am dividing this difference by something. And then depending how big this something is, this difference in the relative numbers could turn out to be really very large or really very small. So in some sense, I am using the units of standard errors in order to decide whether the difference is big or is small. Now, depending on the unit that I'm using here, I will have different answers. Again, so this is the similar problem that I had here. How do I put up these lines so I get the optimal, in some sense, number of calls? So it turns out that this actually is exactly what this general idea of Lima is about. So I would like to borrow strength, meaning I would like to use some of the patterns of these overall gene expressions across conditions in order to figure out what would be a good way of adjusting or deciding upon the units that I would like to use for my gene-wise comparisons. It's very similar, at least up until now, to the idea that I explained or talked about before lunch. So it's really this notion of over-dispersion or dispersion and standard error, variance, and then you somehow are using this idea of a variance when you are looking at the overall pattern of expressions, and then you adjust them to single expression to decide which one of these expressions are more important than others. So we'll try to do this, but first I have to give you a couple more pieces of information. Let me just... So there's a couple different ways in which you can do this. So that's what this slide is really attempting to tell you. And I don't really have time to discuss this in much more detail. In the old days, when we really had much less information about how these various molecular systems are operating, we were using more of the sort of standard non-parametric methods that I mentioned. So I talked about Wilcoxon test. Here I'm talking about randomization tests. These are all okay when you have lots of data and we have lots of people. But when you're looking at systems where you have relatively limited data, or when you have already some basic understanding what is it that you're up against in terms of molecular complexity, there are actually better ways of dealing with various types of statistical noise and standard errors than these standard approaches that have been developed earlier. But if you look at some of the early papers on microarrays and lots of early, not even that early, even nowadays people are using randomization methods, etc. These are okay, and very often there is absolutely nothing wrong with them. They typically tend to have lower power, meaning that it's really hard to see some things even with the relatively simple data sets using these methods. Okay, so they're not horribly bad, but they are somewhat bad. All right, so I would like to discuss the notion of a Lima model, and this is related to some extent to HR. The main difference is that here I am looking at continuous data. So my data, I'm assuming that I'm looking at these signals that are not counts. They're not coming in terms of these counts of different molecules or transcripts that I have sequenced and then aligned. But they are basically coming as some kind of notion of a continuous variable that is either related to the fluorescence of a particular probe, or, as I will tell you later, some kind of scaled version of the counts of transcripts. And depending on your circumstances, and generally speaking, depending on the amount and quality of data, you might go either route, okay, in general. There isn't one good way necessarily of doing it. Ideally, if your data is decent, and if you have lots of confidence in the pre-processing that you have done, it should be true that if you run either one of these methods on a simple example like differential expression, you should get similar answers. But unfortunately, that's not always the case, and it very often is as simple as using different algorithms for pre-processing or for scaling your original signal. Okay? So in some sense, HR is not doing any pre-processing up front. It just looks at straight counts. This method does to some extent. Okay? And again, sometimes it can be good, sometimes it can be bad. That's why we talk about both of them. So I have an RNA. So, I'm switching gear. I'm basically talking about RNA-seq. So sure enough, as soon as the RNA-seq came along, the people who developed these methods for microarrays, they were able to apply them to RNA-seq by applying certain transformations of their data. Maciej is going to at least show us how to do this for specific models. These are the so-called Voom transformations, which are basically turning the types of counts that I was showing you earlier into things that kind of look like fluorescence measurements on microarray. Okay? So the data is different, but the general model works. So generally speaking, what I'm interested in this model when I have a number of different representations. So I have n subjects, individuals, so I have n samples. In each of these samples, I have a large number of genes, which are called G, or tags. I used to call them tags. we are assuming that I was able to transform this thing by this transformation I call the VUM, which, doesn't sound like much. It sounds kind of strange, but that's really how they called it in the early days, VUM transformation. And what it does, it really makes things look like continuous variables. This is kind of the basic model. And then I am assuming that now, because I've already made it continuous, and sort of, by making it continuous, I am also assuming that in some sense I can run the transformation, typically the log transformation on this data, that will make them kind of look like Gaussian data. Not entirely, but at least to some extent it will start looking like a normal, these signals will look like normal distribution. So then, I already told you last time, the normal distribution is also a proud member of this family of generalized linear models with dispersions, but it's much simpler in many ways. Then, lots of things that I was talking about in the context of GLMs can be really looked at over here in the context of regular regression. So in some sense, for the normal member of the GLM family, we are most of the time facing maybe some transformation, but ultimately a linear regression model. So as much as a complicated nonlinear interactions and analyses were possible using this EdgeR software, now once I have VUM'd everything and made it look like a normal distribution, or at least somewhat normal distribution, I will be using the standard regular ideas from the regression models. And that's kind of the gain from doing this and applying these methods, that I basically can use just standard statistics. And statistics for regression has been developed, at least for 450 years or so. So we know a great deal about it, and that's, in some sense, at least for normal data, this is another example of optimal way of doing statistical inference. So I'm assuming that I will have a linear model, meaning that if I take the mean of that particular vector for a single gene, I will end up with a vector of covariates, or the design matrix, multiplied by a vector of coefficients. So depending on the design that I had on my model, exactly like what Soledad was talking about earlier today, I can basically have different sets of coefficients. The gain from this is that I can be incredibly flexible in how do I design my experiment now. Depending on what is it that I really would like to know, what kind of information or what kind of effects I would be interested in estimating, I can basically build very flexible designs that will no longer be looking just at pairs of treatments. They could be looking at multiple different types of treatments. They could be looking at special effects, addition of effects of, as I say, time progression, etc. So there's a number of ways in which you can leverage this in order to get exactly what you want in a sort of prepackaged fashion, because, again, I think Soledad might have mentioned that there is lots of statistical work that was done on sort of general theory of design. So the beauty of this software and this approach is that everything that we know about statistical design theory, you can just apply here. So if you have optimal designs for certain types of data, you can just stick them in here and you will have your optimal design. Okay, so then the assumption is that I have the variance, which is given by this, this type of matrix. All of these things, by the way, are done for a single gene, for a single tag. So in some sense, I'm going to be running 30,000 regressions simultaneously. Each one of them will have its own set of coefficients. And that's how I will be deciding which effects are important. And that's also how I will be picking these genes at the end of the day for myself. So this is the data set that I was using earlier. Again, the similar idea. These were the numbers that we have had. There was 12,000 probes, or there were 12,000 tags that were used. There were 15 individuals with different stages of breast cancer. I think we will be using this data set for the rest of the exercise. So this is something that you guys are already familiar with. At the end, I don't know if I have a, yeah, okay. So I have VUM'd this into basically something that looks like this. So I took my original counts, applied the VUM transformation. Matthew, we're showing how to do this later on. And I end up basically with these things which are now looking kind of like continuous data. And again, it turns out that after the log transformation, they do not look particularly different than at least under, as far as we can tell, they seem to be reasonably approximated by Gaussian or normal variables. So that's kind of what we want to do. Now, what we are interested in, as I said, we are very flexible here with this design. So very often you're not interested in these coefficients. You would like to look at some linear combinations of coefficients. For instance, if you're looking at growth, you would like to look at cumulative growth. So you would like to look at if these coefficients are responsible for some time elapse, then you might be able to keep adding them together and see basically how much the effect of time progression is changing your gene network. Again, if you have data which is time data, for instance. So in this case, for instance, we're looking at different stages of cancer. So in some sense, you can think about it as different snippets, but at different stages of the disease. So you can think about it in a way as a time progression of the disease, which then is reflected in the molecular networks or in various types of molecular processes that are happening. So the idea here is that most of the time I will not be interested in these alpha coefficients, I will be interested in their linear combination. So this is something which is called the contrast in the linear models. Again, I don't really want to spend too much time on discussing linear models, but one of the things that is convenient to deal with is that in these settings you can basically build linear combinations where C is a known constant of these coefficients that you're interested in, and that basically gives you a flexible way, really without no additional cost, of estimating these contrasts instead of the alphas themselves. We're making some standard assumptions about how the estimates of these, so there is a general statistical theory that tells you how do you estimate these coefficients in these kinds of models. It turns out that in some sense these are good methods, and because we are assuming Gaussianity, they are fairly efficient, so we can do this very quickly. That's why it's possible to run these methods in this way, kind of like we did in EdgeR. In EdgeR, we also had fast ways of estimating parameters. If you don't have fast way of estimating parameter in this kind of model, then forget it. You cannot run 20, 30, 50,000 of these models simultaneously. It's just not going to happen, from the practical perspective. So you have to have a model that is going to give you fast answers. So that's part of the reason why we're using these statistical approaches. Otherwise, doing it by hand would be impossible. So we're not assuming that the responses are normal, but we're kind of assuming that they're at least continuous and that there is some way for us to borrow some of the strength from the normal theory. So I have this, if I have an estimate of alpha, then I will create an estimate of beta just by using the same exact set of coefficients that I was using in the matrix C. And then if I know the variance of the coefficients of the matrix alpha, this is very important. If you guys remember, this was something that we struggled with, and we still do in this setting, because that's where we are deciding what SE should look like. Then we will also have this covariance matrix that we can borrow from the alpha coefficients and then adjust it by the C's. Okay, so I showed you this already. So the general idea is, and again, these are sort of, for those of you who have ever taken a class on linear models, these are standard methods. For those of you who have never seen it, it probably doesn't mean much. But the idea is that we are assuming certain things here. So in order to turn these estimates quickly and be able to do the calculations that we would like to do, we're basically assuming, okay, approximately, and that follows from the general statistical theory, these estimates of betas, which is what I want, they are, if I know what the true beta was and what the true sigma or the variance was, then they kind of look like normal distribution. So here, I'm assuming, even though the data might not have been normal, I'm assuming that my estimates that I get are approximately normal. And then the same thing works for the S, which is the estimate of sigma. So I'm making these assumptions, where dg is the number of residual degrees of freedom for the linear model, so it kind of depends on the number of covariates that I had, et cetera, et cetera. So then under these assumptions, I have this statistic T, which is basically, really, what I have shown you over here already. Okay? It's just I use a little bit more fancy notation, and I have shown you more specifically that I'm not looking at the gene-gene interactions, but I have now turned this entire problem into the problem about coefficients of the linear model, okay? But each of these linear models is running on a specific gene, so it's really the same problem. Okay, so what I'm looking at is this thing over here, but now I still have the problem that I had on this early slide. I really have this SG and VGJ. Okay, so this is all good, but this is gene-specific, and this is for the jth beta coefficient, so this is a number now. How do I figure out what this thing should be? You know, I have some estimate of it, but if I only saw 2 or 3 patients, then this sort of estimate based on the number 3 of data points is probably going to be horrible for me. So that's what I build, what we call a hierarchical model. So we basically will use the information that we are borrowing from other transcripts or tags in order to figure out what this SG should look like. And in fact, what we will end up with in a moment is a version of this thing which is called a moderated t statistic, which will take this, and again, you know, I always like to at least bring to your attention that there's lots of similarities. Lots of these ideas are really very similar. So this moderated t statistic that I will show you in a moment has a lot to do, at least conceptually, with this notion of shrinkage. If you remember, I was telling you about this idea of shrinkage of your over-dispersion parameter when you would, on one hand, use the particular over-dispersion parameter for a tag, and then you would mix it up, if you will, with the overall estimate of the shrinkage for all of the tags together. This is exactly what this statistic is doing. It's taking the value in the denominator that comes from its own particular tag across all of the subjects, but then it also adjusts it for the overall patterns of all of the tags altogether. And that's basically how this moderated t statistic is decided. It's defined. Right. So let me move on. So that's how it looks like. Okay. So again, I don't really want to go into too much detail. I've already spent, I think, enough time. I would like to give you a chance to experience this. But this moderated t is basically so up here, and then the v is basically what it was, and this s is now s wave. Well, now I am adjusting, so this s0 is sort of overall dispersion across all of my tags, and sg is the dispersion for the particular tag, d is the number of degrees of freedom or the number of covariates that I have in my model for the tag-specific model, and d0 is the total number of transcripts, in this case, that I have. So what it does, again, it achieves this notion of shrinkage and smoothing. So you're sort of shrinking to the overall pattern, the pattern of your particular gene expression. And what people have shown is that this is very effective in trying to give us a way of deciding which of these differences in my t statistic at the end of the day are high or low. So basically how close this thing will be to 0, because that's ultimately what we're interested in, versus, you know, where this number is really different. So everything about this number is related to by what do I divide it. If I divide it by a very small number, it's going to be very large. If I divide it by something very large, it's going to be small. And that will basically make the difference in terms of calling something close to 0 or not. Okay? So the general idea is that we have this moderated t statistic, which really, in the continuous setting, in the setting of the linear model, is realizing the same principle as our shrinkage estimate of overdispersion was in the case of my previous model. So the marginal distributions, so there is a number of ways in which I can then try to adjust these t statistics in order to test for whether or not the true coefficient beta is 0. Under that assumption, that statistic has certain distribution, and I can use this to calculate the p-values. So at the end of the day, what we do, we calculate this O statistic, or log O, I think people call it B, which is basically the odds that the particular gene is differentially expressed. So we're basically taking this thing one step higher, and instead of looking just at the straight difference, we actually calculate log odds of the event that particular gene would be differentially expressed. Okay? So if the odds are 50-50, so if the probability is 50-50, then the odds would be 1, and then this thing would be 0. Right? So log of O close to 0 means that there is no difference, really, or that statistically that particular tag doesn't seem to be likely to be differentially expressed. Anything which is a large number, either positive or negative, will indicate that that particular tag is likely to be expressed. So this is another way, in addition to the p-value, of evaluating your differential expressions in the context of this really, really general model that I have just outlined. So one last thing that I want to mention, so this is basically the Lima model. This is how this Lima stands for linear model for microarray analysis or something like this. So that's the name. But really what they came up with this log odds type of statistic that they are using, which is based on the shrinkage idea and the idea of borrowing strength across transcripts in order to be able to identify things which are differentially expressed. Now, how do we actually make the connection with of something like this with the false positive or some kind of false discovery rates? So again, I don't really want to spend too much time on this because I've already overextended myself time-wise, and I would like for you to try it on your own. So let me just say the following. There's several different ways in which people are trying to make sense out of this notion of differential expression and false discoveries. One way, the one that seems to be fairly successful, there's a couple others, is you can think about it in terms of the potential number of discoveries that you have across all of your genes. Okay? So think about it this way. You have M genes. For real, M0 of them are differentially expressed, and M1 of them are not. So if you are interested in identifying the M0, then you basically will have some guess for what that should be. In reality, you will have the following 4 cases. You will have true positives, the ones that you correctly identified. You will have false negatives, the ones that you didn't catch, even though you should have. And then you will have the ones which are false positive, the ones that you identified but they shouldn't be there, and the ones that you did not identify, and rightfully so, because they had no business being in your list of discoveries. So here and here you're making the correct decision. Here and here you're actually making a mistake. So now, depending on how interested are you in these 2 types of errors, you can basically have different ways of trying to handle the error in general. So one way, at least originally, nowadays I think it's becoming more popular to look at both of them simultaneously, but originally people have really been concerned about false discoveries. You don't want to have too many genes that don't seem to be necessarily expressed, but you are, in your analyses bringing them up and then trying to test them in the lab, and you're spending lots of resources on chasing something that from very beginning was very dubious. So you want to have a limited number of these false discoveries among your discoveries. So that brings, again, there's several different ways in which you can define it, but the one that I've highlighted and lots of people seem to be using nowadays, even more so than in the past, is this notion of false discovery rate. That's what we already briefly talked about. The idea is really very simple. This is Fn, false negative, divided by the total number of discoveries, R. Okay? So if I have had total number of discoveries, which is this number, and if I took this as the denominator against false negatives, then it basically gives me the false discovery rate. So this is really the number of incorrect discoveries among all of your discoveries. And you would like, in various ways, you would like this number to be small. You would like to keep it at some reasonable level, 1%, 5%, what have you. Turns out that there is a straightforward way of doing this thing relatively straightforward, within the context of the Lima model. And as you will see, it makes a big difference, at least in the example that I will show you in a second. The method is like this. So I can calculate p-values based on these moderated t-statistic. And once I have p-values, I can order them up. So I will have 20,000 of these p-values calculated for each and every tag. And then I will basically follow the procedure like this. I will take the last gene, the one that has the highest p-value, and I will basically multiply it. I will blow it up by this factor. This is called an inverse harmonic average, by the way, but it really doesn't matter. This is just the factor that sort of comes out of the consideration of what this false discovery is and how do you bound it by something reasonable. And then you multiply it up and you make it your p-wave. So this is your new p-value adjusted. And then you take this one, you remember it, you go to your next p-value, and then you take the minimum of that adjusted p-value against, again, the little bit blown up the p-value that's coming from your next gene, etc. And you keep doing this until you're done at the very, very lowest level. So what it does, it basically, it blows up by certain factor the p-values that you would get out of the analysis of these tag-wise expression. Why is it that we're doing that? So you can imagine that in real world, when you have these tag-wise tests, just because of the fact that most of the time we're building into these tests some level of type 1 error, so basically 5% of discoveries would be purely by chance. Then, if I am running, 100 of these tests, then in 5 cases, even if there was no difference, I would just purely by chance observe that there were differences, right? So this is basically this is called multiplicity adjustment, and the idea is that if you're running lots of tests, and each of these tests you have a small chance of being wrong, even if there is absolutely no difference and you shouldn't be, then basically this cumulates up. These things are adding up. So basically your chance of being wrong in the large number of discoveries, it's cumulative. It adds up, and eventually it will add up to something that you're losing control of. So what this process with blowing up your p-values is doing, it's basically keeping this under wraps. It basically allows you, as you're moving along with your testing, it adjusts these p-value larger, so you can basically keep adjusting for the fact that you're running these tests multiple times. At the end of the day, whatever p-value, adjusted p-value is below 5%, you would declare that particular p-value to be significant, and the gene or the tag which is associated with this, you would declare to be differentially expressed. So let me show you how this works. And this is an example that I picked up because it's actually sort of mind-blowing in that sense. We'll discuss how to do the fits and the top tables in a moment. This is part of the Lima software. But look basically how it works. So I did the log of the fold change for this genes under 2 different conditions. I don't remember. I think I was comparing stage 1 and stage 2. I had the average expression. I had log fold change, so negative 1.5. So about, this is log 2. So this basically, so it indicates that there is potentially some difference. This was the value of this moderated t-statistic. This was the original p-value, very tiny, 0.0001, so less than 0.1%. If I adjusted a p-value, it basically looks like 1. So after I've done all the adjusting for the 12,000 of these expressions, this p-value, this differential expression just goes away. It's no longer considered to be interesting. It's basically saying, look, you've been running so many of these tests that if you have something at this level, even though technically, for this particular gene, it is a significant finding, overall, in the context of all of your finding, it is not. Okay? So you basically can see how this works here, and we tried it also. Again, I picked up some of these things purposefully just to show you how this works. So if you don't know about adjusted p-values, if you just run this Lima software and you get excited about some of these B statistics, oh, they're large, it's -4, so potentially, I'm finding lots of cool genes. But if you do the adjusting and it sort of looks like, well, no, you're not, then you at least know that it's probably something that you have to spend some time thinking about and then potentially reevaluate whether you get something useful out of this or not. So this is sort of, in a nutshell, the way in which we analyze differential expressions. The model that I explained to you is very general. It can be used for a variety of different things. Here with MACHE, we have only really tried it on these 3 different stages of breast cancers, looking at the differences between stage 1 and 2 and stage 2 and 3, but I could have done it in a much more fancy way. I could have looked, for instance, for genes that were initially involved in the creating of the early microenvironments in stage 1, but they were not involved in stage 2 and stage 3. So I can try to build these different types of hypotheses, then I can stick into my linear model and then try to run them and find the subsets of genes that would be relevant to what I'm trying to find. So the beauty of this approach, and really the reason why I like it, is that it is incredibly flexible and it allows you for a variety of hypotheses that you can test really on the fly just by changing the design. And it really doesn't cost you anything in terms of increased computer times or some additional manipulations of the data. Once you read your data into Lima, once you've zoomed it, you have basically ability to relatively easily adjust for different types of questions that you would like to ask. So we'll try to run some of these examples to give you guys a chance to play with this, and let's see what happens, okay?
Run the analysis. So whoever join us after lunch, please download from the box differential expression folder, and you will find the Lima files. For people who were with us earlier, please open the Lima files. And again, you got the Lima assignment. So basically, this is a kind of PDF file with things we would like you to do and try to figure out by yourself step by step based on the lecture slides and my hands-on slides. And you also will have a code just to review the whatever you are trying to do. And right now, I will just quickly go through all the process step by step, and then I will go and I will walk between rows, and we can basically work together and do the analysis. So basically, we are starting with whoever haven't installed the package. This is the code how to do it. So first you call source bioconductor, and then using bioscilite function you install Lima. For everybody who installed Edger before, this package should be installed together with the previous package. So first we read in the data set. So basically you use yes? Of course. So first you run function read CSV, and this time you will use Lima dataset CSV file to RNA counts. And the first column would serve as a row name, gene name information that you would set as row names. And you delete the first column using this command. And this is what you're supposed to see after calling the first 6 rows of and first 7 columns of the data set. Now we define the contrast table. So we'll have stage 1, stage 2A, and stage 3, and we will compare 3 stages of the cancer in this analysis. And so kind of to see how it aligns with your sample names, you create this cancer code data frame where you got the call names of RNA counts that are the sample names and stage identifiers. And you factorize the identifiers of the stage of the cancer and create a design matrix that should look like this. And you create contrasts. Basically, you are telling what would you like to compare, which groups you would like to compare with each other. That should look like this. And the next step is a VUM transformation of your count data. And you plug in the design matrix, and this is what you should obtain. So this is very similar to the matrix you've seen during the lecture. And the Y object is the Lima data objects, and similarly to Edger, you would see various elements of this object that contain all the necessary information to run the analysis. And then I would ask you to create the MDS plots. So basically, this is this plot which compares overall expression profiles of, in this case, top variable genes. And here you would replace this. So since we got all this like sample names are not very informative. So in this stage, you would replace the sample name with the stage information to see whether you got any like patterns between the similarity patterns between the expression profiles within the stages. And here are the most important part. So you run differential gene expression analysis, calling lm-fit and eBias function from Lima package. And you get differentially expressed genes from the Lima data objects using a top table function for coefficient 1. So this would be stage 1 and stage 2a, coefficient 2 and 3. And then I would ask you to write data to the files. So basically you call a top table function with the number of genes that would be equal to the yes. So these are the functions. So this runs the linear model fit. And eBias runs this Bayesian statistics. So basically, it adds information to the Lima data object that you call by top table as this table of differentially expressed genes. And then you just write to the files the results of analysis for each coefficient. And do you have any questions? If not, please start and let me know if you got any questions.